Welcome everybody, and thank you for attending our today's webinar. So first I would like to introduce myself. My name is Oliver Wehmeier. I'm a cell biologist by heart and by training, now mainly running ACCELLerate, a company focused on the custom manufacturing of analytical cell banks. And I would like to give you an insight on an approach which has been widely adopted now for different kinds of cell-based assays, and this is the use of cells in an assay-ready format, which means that you use the cells basically like in a reagent. You take them directly from the cryo stock into your assay plate without propagation or cultivation of the cells up front. This is an approach which has first been introduced in the drug discovery field in high throughput screening back in 2004, and has now been widely accepted and adopted also for bioassays because it has actually a lot of advantages. I assume that you're all, or most of you, working in the field of cell-based assays, so you probably all know that there are a lot of factors which have an impact on the quality of a cell-based assays. Of the cell culture and the conditions of the cells, and thereby also on the cell assay, starting of course from the cultivation of the cells, the handling from user to user, and also the consumables you're using. In particular, the serum can have a significant impact, of course, on the quality of your cell culture and the outcome of the cell-based assays. But also the other consumables, culture vessels, pipettes, the equipment you're using, the incubators, how is the carbon dioxide set, how is the humidity, all this has an impact from a more technical part, but also from the cellular level. There are various factors which influence the culture, which is the stability of the cells, basically the aging, the passage number. The more you passage the cells, the more likely it is that the cells might change their properties. And of course also sterility and identity, absence of any kind of contamination, even cross-contamination from other cells, and also the storage of the cells has an impact. So if you think about using cells in a classical way, means you keep them in a maintenance culture in small tissue culture flasks. The day before the assay, you harvest the cells and dispense them into your assay plates. All these factors will have an influence on your cell-based assays. So there's a lot of variability coming into the assays by different handling, by different lots of material. We have a passage drift of cells and always, of course, a risk of contamination. I will give you two examples on this. This is an recombinant reporter gene, luciferase reporter gene cell line. And if you grow the cells in an exponential healthy way, always keep them subconfluent. So this is the red curve here. You will get a very nice signal to noise and very nice assay window. If this cell line is grown too confluent, the signal is lost. And strikingly, this is not only the case if they grow confluence directly before harvest, this also is the case if they have been too confluent anywhere during the expansion of the cells. So this cell line really needs a long time to recover. This is to demonstrate that confluence is a critical factor in your continuous maintenance culture, and even a single day of overconfluence you can run into problems with your assay. The second example is about stability. This is a recombinant cell line expressing a surface target. And if you look at these cells, the target expression by flow cytometry, you can see at passage 8, this is the gray is the isotype control. You can see a nice expression of your target by flow cytometry and the fluorescence signal. If you expand these cells further and grow them further at passage 21, you see another peak. So there is an increasing amount of cells which lost the strong expression of your target. So this is something which indicates that stability or aging of the cells can be a critical part which you need to observe. So again, there are a lot of factors which have an impact on your cell culture, and there is a smart solution to this, and this is preparing all cells at the same time. And this is the approach of using Assay Ready Cells. The idea is to expand the cells to a large quantity, for example, in these cell sticks here, and then harvest all the cells all at the same time and cryopreserve them at a very high quality functional way, so that you then use these aliquots, pre-qualify. You can pre-qualify these aliquots and say, okay, these aliquots are working in my assay. And as long as you store them properly, you can use single aliquots of this cryopreserved assay ready cells, basically like a reagent, thaw them, dispense them into your assay plates, and then you're ready to go. And by this approach, all these variabilities you have from cell culture are kind of standardized by this large batch production of the cells and are excluded from, or at least standardized in your cell-based assay. This is some data to demonstrate how precise and accurate the use of Assay Ready cells are. On the left, this is a TNF-alpha cytotoxicity assay performed on L99 Assay Ready cells. So L99 respond with a toxic, TNF-alpha is toxic in a dose-dependent manner. And L99 cytotoxicity assay is a common used bioassay to determine the potency of the TNF-alpha preparation. And you can see here 6 independent runs, which means these are 6 independent vials of assay-ready prepared L99 cells, which have been thawed and then used in the cell-based assay without cultivation, even without counting of the cells. Just the entire quantity of cells is dispensed into the assay plates, and then the cytotoxicity assay is run with increasing amounts of TNF-alpha. And you see that you have a very high precision when you compare these individual runs of the assay. And the use of Assay Ready cells is also quite accurate. We have analyzed the potency from different endpoints, different cell lines from different projects, and compared the results from when we used continuously cultured cells with Assay Ready cells, which had been used instantly after thawing. And you can see here that we have a really great comparison or regression when we compare the continuously cultured cells with the Assay Ready cells. They only have a single outliner where it was difficult to obtain this. And with all this experience since 2004, they only had a handful of projects where it was a little bit more difficult to obtain that. But in general, the use of Assay Ready cells can very well applied for different kinds of cell-based assays. So the question is, what is actually the difference between preparing a regular cryo stock of cryopreserved cells and an Assay Ready cells? And the answer is, in fact, there is no real difference. The difference is that you have to keep in mind that you want to use the cells in Assay Ready format, that you have to cryopreserve them at a highly functional level. So there's no secret ingredient, there's no proprietary technology in preparing these Assay Ready cells. It's simply the know-how, how to handle it and to do it right. So first of all, the expansion of the cells needs to be very controlled. The cells should not be overgrown, and we should harvest them in an exponential growth phase. Always keep in mind that the cells, when you harvest them, are in the state when you use them in the final assay. There's no way that they recover from the cryo passage. So if we haven't treated them well during freezing, they will not work when you use them in the assay. The most important point, of course, is the cryopreservation process itself. So there's always a need to optimize the cryopreservation media. There's definitely no one-fits-all media which work the same for all cell lines and all cell types. And also the optimization for the cryopreservation protocol it has a significant impact. I will come to that later. Finally, of course, also the storage of the cells in liquid nitrogen is important, and the thawing and preparation of the assay ready cells for your assay is critical as well. Particularly the storage, you should of course keep them in the vapor phase of liquid nitrogen and not take them out too often, which definitely will increase the performance of the cells. Coming to these two most critical points of cryopreservation media and protocol, this is an example which we use for quite a number of projects, not for all. As I said, there is no one fit all, but we have developed a serum-free freezing medium with the rationale that we replace the serum, remove the serum to get rid of any growth factors to avoid any conflicting signals while the cells are frozen. Replace glucose with higher molecular sugars because the cells don't need any glucose while they're frozen. The osmolarity has been adjusted in this freezing medium because the active sodium and potassium pumps are not working anymore, so there has to be a different osmolarity of the medium. And very important as well, we always reduce the amount of DMSO for cryoprotection to 5%. The still commonly used standard of 10%, to our experience, is way too high for most of the cell lines. We can even go lower than 5% or replace DMSO by alternatives as well. Talking about cryopreservation, which is the second important factor on high quality freezing of cells, is using a controlled rate freezer. It's not only that controlled rate freezer controls the cooling rate, which is commonly 1 degree per minute, but again, not necessarily always. We have a number of cell lines where much quicker freezing is of advantage, of benefit. There's another difference between a controlled rate freezer and a classical isopropanol freezing container, which is frequently used in laboratories. The point is, if you use this, which applies a cooling rate of 1 degree per minute, this is fine. If you look at these temperature traces here, the temperature is decreasing below 0 degrees, and then the crystallization occurs. And at this very moment where the medium actually freezes, where crystals occur, there's latent heat is released, and this release of latent heat increases the temperature in the sample and have a negative impact on the quality of the cells. By using a controlled rate freezer, not only the temperature decrease is more controlled, a controlled rate freezer also compensates this increase in temperature, so this release of latent heat, by blowing liquid nitrogen into the chamber at this critical moment of crystallization. And by this, the freezing process in a controlled rate freezer is superior to any kind of mechanical freezing device or isopropanol container freezing devices, for example. And this makes a big difference. Of course, you can prepare standard frozen stocks in an isopropanol container as well, but those cells will usually not have this high immediate functionality which you can obtain in a controlled rate freezer. I have one example here for you, which is THP1 cells. THP1 cells, most of you might know them. It's a human monocytic cell line which grows fairly well in suspension. But if you recover THP1 cells from a standard frozen stocks, cells usually need a while to recover. And I can demonstrate this here in a propylium iodide staining of the cells. On the left column, you see cells from a continuously passage culture, and you hardly see any PI positive cells. So this is a really healthy culture. In the middle column, these are cells which have been recovered from a standard cryostock of THP1 cells. Immediately after thawing, the cells are of high viability. But 24 hours later, there is an increasing amount of PI positive cells also after 48 hours, which indicates that a lot of cells are dying after thawing from the frozen stocks. And if you know THP1 cells, you will confirm that these cells usually take a week, sometimes even longer, to fully recover from a frozen stock, and you can use them so that they fully enter proliferation. With this optimized freezing procedure, we have established the different freezing media in individually optimized cryopreservation protocol for THP1 cells. We were able to overcome this crisis the cells are going through. So even after 24 and 48 hours, there's no significant increase of PI positive cells. There are a few, but as long as not as much as you see from a standard process. And if you use these cells in a proliferation, or in this case an anti-proliferation assay, you see that the Assay Ready cells, which have been prepared with our improved protocol, which is the red curve here, are almost as good as the cells from a continuous culture, the green curves. While cells which had been frozen according to a standard protocol display a reduced assay window because the cells do not immediately proliferate as good as a continuously culture from an exponential growth rate. So you see that there is a lot of room for improvement in cryopreservation so that the cells become really functional when you thaw them and immediately enter proliferation and do not go through kind of a lag phase after thawing. So the benefits of using Assay Ready cells is quite obvious for most of the parts. So first of all, you save some laboratory resources. We did some calculations on that. Depends on 10 to 30%, depending on the assay type and the cell type, of course. Simply by using Assay Ready cells, which have been prepared in a large homogeneous batch, you reduce the work compared to a continuous maintenance culture. The most important part, in particular for the use in bioassay, is that you have a homogeneous cell bank of pre-qualified cells which reliably perform. You can pre-qualify these valves, and this can improve the precision of your bioassay very much and decreases the risk of an assay to fail. Assay Ready cells are instantly available, so you very much increase the flexibility of your bioassays. You can basically decide the day before how many valves you want to thaw, how many assay plates you need to run, and this is of course much more flexible, and you're working with consistent cells even after use. As said, as long as you store the cells properly, the cells reliably perform the same way even after use. Finally, Assay Ready cells are convenient to use. If you think about working together with CROs or external partners performing your bioassay, then the transfer of the cultivation protocol is usually the most critical and difficult part. And if you can exclude this by simply providing Assay Ready Cell Banks to your CRO partner, this makes your life much easier when working with external CROs. This is an example where we have applied Assay Ready cells, pre-qualified Assay Ready cells in a TNF-alpha neutralization assay, which is the commonly used or one of the most frequently used assays to determine the potency of TNF-alpha neutralizing antibodies. So we tested here adalimumab and etanercept, and again have run the assay multiple times with the Assay Ready L99 cells. Again, one single aliquot has been thawed, dispensed in the assay plates to run the neutralization assay, which means that the cells are challenged with TNF-alpha, which would usually cause them to die. And by an addition of an increasing amount of neutralizing antibody, the cells can be rescued from the toxic effect of TNF-alpha. So you see that increasing amounts of antibody increases the viability of the Assay Ready L99 cells. And again, you see a very, very high reproducible and precision of the assay when you're using Assay Ready cells. At ACCELLerate, we have established a process to prepare custom cell banks of Assay Ready cells. We have capabilities for large-scale cell expansion using the cell stacks or hyperflasks. Then we have an automated dispensing system, which allows us to very accurately fill 1,000 cryovials within a very short time of 30 minutes. And then, as I already demonstrated, we are using a controlled rate freezer with individually optimized freezing protocols for different cell types to really obtain this high quality cryopreservation of the analytical cell lines. And then, of course, we store the cells in vapor phase in a high quality condition until they are released and sent out to our customers. We can prepare suspension cells and adherent cells and prepare cell banks of up to 1,000 vials, which is the limitation of our control rate freezer. All cells are prepared in homogeneous batches, so all cells are harvested at the same time. And we can prepare custom aliquots from 1 million cells, which is more at the lower end for bioassays where you do not need that many cells, but up to 100 million cells, which apply only for smaller cell lines. But if you think about virus inactivation assays, for example, then a high cell number is required for bioassays. Yeah, that's basically an overview about the use of Assay Ready Cells and how much they can support your bioassays. Feel free to ask some questions, write them in the chat, and I'm happy to respond to it. Or if you have any more specific questions, just contact us by email. Thank you. Is the cell number of frozen cells fixed? It can be flexibly adopted. As I said, when we do this as a custom production, we can do it from 1 million to 100 million cells. 100 million can be a thick paste if you have larger cell lines like CHO or U2OS. If you think about a smaller Jucat or THP1, 100 million cells per standard 2 milliliter cryovial works fine. Is there a limitation of the number of cells per vial? This is a kind of a good question. Is this from an economic perspective? Yes, because then the preparation of the cell banks with such a low quantity of cells it's sometimes better to discard the remainings of the cells rather than have this small quantity frozen. I would say yes, there's also a physiological or biological limit. To our experience, the cells recover not as good anymore when they're frozen below 500,000, below 500,000. So I would not go below 1 million per milliliter. There's a question, have you seen any cell types at Assay Ready Cells which does not work well? I would not say that there are particular cell types which does not work well. So there are cell lines which are already difficult to cultivate and which are really hard to cryopreserve in general. Those are more tricky to prepare in assay ready format, of course. In particular, very slowly growing cell lines can be challenging, but so far we have been able to achieve this as well. It's more on if you think about target expression or something like that, this can be tricky. So it's more a question about the target which you're looking at than the cell type in general. Can all cell lines be prepared? Yes, most of them. Is there any cell patent issue if you sell the product to pharma companies? No, there is no proprietary technology behind the preparation of assay ready cells. Of course, the cell lines itself, when we do this as a custom production, we usually receive the cell line and master valve from our clients to prepare the cell bank. If you resell it, there's no proprietary technology included in the preparation of the cells. There's a question, are you aware of any bioassays which are using your cells in the release panel of FDA approved molecules? Yes, we have a couple of customers who are using analytical assay ready cell banks prepared by us for product release in CMC. Have you seen any increase in adherence? I'm not sure what that means, to be honest. So do you mean that the cells heal quicker when they're prepared in that way? So our aim is, of course, to cryopreserve the cells in a way that they adhere as quickly as good as possible after thawing, and this is usually something which runs in parallel with the quality of the freezing process. If the cells are freezing properly, they usually also adhere quicker than cells which are not cryopreserved at that highly functional level. The batch-to-batch consistency, yes, this is also a good question. We have established, of course, a master cell bank, working cell bank, Assay Ready Cell Bank regime which allows us to prepare individual batches of Assay Ready cells according to the same passaging regime as the other batches. And by this, the reproducibility from batch to batch is usually very high, which means that we are almost come out with almost the same passage and using the same medium, the same materials, which then results in a very good consistency and reproducibility from batch to batch. The question on the costs, of course, this is a certain price on preparing custom preparation of Assay Ready Cell Banks. This very much depends on, of course, the size of the batch and the size of the aliquots. So we have quite standard and fixed pricing per vial, and the more vials you order, the larger the batch is, the lower will be the price. And of course, the more cells are in a single aliquot, the higher will be the price. And we have these kind of standard prices which cover most of 90% of all the cells. Only if there are cells which really need special treatment, like induction or very expensive growth factors or supplements to the medium, there might be a surcharge. So if you have a concrete request, just send me an email and I'm happy to provide you with a concrete quote. Do you test the cells? If the cells work before releasing? Yes. So the quality control we apply to our custom Assay Ready cells includes, of course, test on viability and cell count and cell recovery on the cell level. Then all Assay Ready Cell Banks we release are tested for sterility and absence of mycoplasma, of course. For sterility testing, we inoculate indicator process. Mycoplasma testing is done by PCR, and we also test for the identity of the cell lines, which means we perform STR analysis, which is externally done for human cell lines, and have established a species-specific PCR to exclude any cross-contamination from non-human or other species cell lines. And we also perform functional assays, which depends, of course, on the individual cell line we receive from our customers. This can be flow cytometry to document target expression, but this could also be reported gene assays, for example. All the batches will be delivered with complete batch record, which document individual handling steps of the cells and passaging steps of the cells, and a certificate of analysis which summarizes our quality. Okay, so thank you all for your attention, your interest. If you have any further questions, don't hesitate to send us an email or contact us through our webpage. Thank you. Have a good day. Bye-bye.